Hello again, this is Andrew Becker. Prove it to you. And for my reproductive systems portfolio, I decided to focus on the process of spermatogenesis, or the creation of sperm within the male reproductive system. We'll start by taking a look at the testicles or testes. These are the internal male genitalia. During fetal development, they actually form inside the abdominal cavity and migrate out through the inguinal canal and eventually end up in the scrotum. They are covered by a fibrous sheath called the tunica vaginalis, and inside we find these lengths of coiled tubules, the seminiferous tubules. These occur in pockets separated by septa. There are about 250 to 300 of these. And these are where the spermatogenesis takes place. Once the sperm are released, they are flushed out of the seminiferous tubules through the rod of testes, and then enter the epididymis, where they complete their final maturation. Eventually, they get pushed out of the epididymis through the vas deferens, where they eventually meet up with the urethra and are ready for ejaculation. So, in order to get a good understanding of the process of spermatogenesis, we really need to first look at the wall of the seminiferous tubule. There are three layers here: an interstitial layer, which I couldn't really show in this photo. That contains Leydig cells, which are endocrine cells that secrete testosterone, as well as the capillaries and lymphatics that nourish and drain the tissues, as well as these peritubular myoid cells. This is a specialized form of smooth muscle. When it contracts, it helps move the sperm along the length of the seminiferous tubule. But it also has some very interesting paracrine functions, which we'll talk about towards the end. It abuts a basal lamina. We can get my cursor back here. There we go. Basal lamina, which anchors the epithelial tissue above it. And this is where all the action really takes place. The epithelial layer contains the spermatogonia, the stem cells, the germline that kind of give rise to the male gametes. Here we see the spermatogonia differentiating into primary spermatocytes, which then undergo meiosis to produce secondary spermatocytes, and ultimately four sister haploid spermatids, and that will go on to become spermatozoa or sperm. And the workhorses of this whole process are these Sertoli cells, the actual epithelial lining of the ducts. You can see the nucleus down here by the basal lamina, and it extends all the way up to its apical membrane. These form very close interactions with the developing spermatocytes. You can see how nestled in these developing cells are. And the Sertoli cells are really responsible for shepherding them along and into the lumen, and they secrete a whole bunch of different growth factors and cell signals that really guide their differentiation. They secrete an androgen binding protein, which helps concentrate testosterone in the lumen. It makes it less lipophilic and more easily dissolve there, and also forms these tight junctions right here, and these will actually dissolve and reform as the spermatocytes kind of progress through them, but they help form this blood-testes barrier. It's analogous to the blood-brain barrier, and that blocks the immune system and other potential contaminating compounds from interfering with this very delicate process of gamete development. So here we'll take a look at the actual phases of spermatogenesis without kind of the interference of all the other Sertoli cells and other things going on around it. We start with spermatocytogenesis, the creation of a primary spermatocyte from the spermatogonia. And this is just kind of general regular mitosis, where a diploid cell doubles its DNA and undergoes division to produce two daughter cells. One of these cells will remain spermatogonia and replenish the germline, and the other one goes on to become a primary spermatocyte. That primary spermatocyte will then undergo spermatitidogenesis and start going through meiosis. So it starts by doubling its chromosomes in preparation to divide, and then it undergoes meiosis 1, which produces two secondary spermatocytes. These will go then further divide in meiosis II to produce 4 haploid spermatids. And then these spermatids continue their maturation process, undergoing some very drastic changes in morphology, to become spermatozoa in the process of spermiogenesis. During spermiogenesis, the spermatids undergo a number of changes, very drastic changes, that alter their morphology into that iconic sperm shape. This starts in the Golgi phase, where the Golgi apparatus starts building up lytic enzymes that will eventually help the sperm break through the matrix surrounding an egg follicle and complete fertilization. This is also the stage where the DNA of the spermatid gets packed up and loses its kind of transcriptional ability, becoming inert. And this is followed by the cap phase, where the acrosome forms from that Golgi body on the anterior aspect of the cell. Following that, microtubules start extending out from a centriole and out the back of the cell. This will eventually form the flagellum. And the mitochondria that kind of power the motor complex in this middle portion here migrate towards the center in preparation to form that little cellular engine. Finally, during the maturation phase, the spermatid expels all of those excess organelles and cytoplasm into this residual body. That is then phagocytized by the Sertoli cells, and the formerly spermatid, now official spermatozoa, is ready to be released into the lumen. It is officially considered a sperm at this point, but it still needs to undergo maturation in order to become viable. That occurs as it flows along the seminiferous tubule, but is really kind of perfected once it reaches the epididymis. The cells lining the epididymis secrete proteins that further guide the maturation. These include modifications to flagellar mechanics, as well as a lot of different cell membrane modifications. It actually makes it more negative and changes the lipid rafts around to kind of make it more specialized for entering an egg cell. The entire process from the first division of the spermatogonia to complete sperm takes approximately 76 days. The majority of this takes place in that epithelia in the seminiferous epithelia between the Sertoli cells. That's about 64 days. And then the spermatozoa spend about 12 days in the lumen of both the seminiferous tubules and the epididymis. As you might imagine, hormonal control over this whole process can be pretty complicated, so I decided to break it down into both the endocrine and paracrine sides to try to simplify it a little bit. The two most important hormones that kind of foster spermatogenesis are testosterone and other androgens, but mostly testosterone, and follicle stimulating hormone from the anterior pituitary. We'll start with the hypothalamus, which releases gonadotropin releasing hormone. This goes on to the anterior pituitary, stimulating the release of two other tropic hormones, luteinizing hormone, which will go on to affect the Leydig cells, causing them to secrete testosterone, and follicle stimulating hormone, which acts directly on the spermatogonia and Sertoli cells and prompts their production of gametes. Testosterone will have a negative feedback effect on gonadotropin releasing hormone and luteinizing hormone, and we think it also has an effect on follicle stimulating hormone, but there are other pathways at work there, so it doesn't perfectly correlate. On the paracrine side, we see paracrine activity within the hypothalamus, since GnRH is regulated by a polypeptide produced there called kisspeptin. Obviously, the primary site for paracrine signaling occurs in those Sertoli cells, those nurse cells that we talked about earlier. These actually contain the testosterone receptors. The spermatocytes themselves don't actually carry any, so these are the ones that are kind of responding to the hormonal signals. It can modulate the strength of this signal by converting testosterone into dihydrotestosterone. This is a much more potent form, and it kind of helps amplify that signal, or by extension, if it decides not to convert it, mute the signal. It also releases two other hormones, activin and inhibin. They function in a paracrine fashion by activating, in the case of activin, and inhibiting testosterone in the Leydig cells. But they also have an endocrine effect as well, as they go on to either suppress or further activate follicle stimulating hormone in the anterior pituitary. Interestingly, these are expressed in a lot of other tissues throughout the body, so their role in cell maintenance is probably a lot more diverse than what we're talking about here. And finally, the peritubular myoid cells, those specialized smooth muscle cells that abut the basement membrane in the seminiferous tubules. As I mentioned earlier, they do have some interesting paracrine functions. They're still kind of a mystery, but we do know that they possess androgen receptors, meaning they're sensitive to testosterone and compounds like it. And they also express growth factors and intercellular signaling molecules that are known to act on the Sertoli cells. So this indicates that there is kind of crosstalk between both the Sertoli cells and these myoid cells. Finally, I wanted to end with this really nice diagram from the Silverthorne text that does a good job of tying together both the hormonal activity and the cellular activity that goes on in spermatogenesis. Here we see the wall of the seminiferous tubule. It's the interstitial layer, the Leydig cells, the basal layer and the abutting myoid cells, and the epithelial layer with their Sertoli cells, spermatogonia, and developing spermatocytes. So as we talked about, the whole process starts in the hypothalamus, which secretes gonadotropin releasing hormone, which in turn acts on the pituitary, causing it to release both follicle stimulating hormone and luteinizing hormone. That luteinizing hormone will have a direct effect on the Leydig cells, causing them to secrete testosterone, which goes on to the other peripheral tissues of the body, causing various effects there, as well as entering the lumen of the seminiferous tubule, where it helps further the maturation of the sperm. Testosterone has a negative feedback effect on both gonadotropin releasing hormone and luteinizing hormone. On the follicle stimulating hormone side of things, we can see that it has a direct effect on the Sertoli cells, stimulating the production of more gametes, more sperm. And it has a negative feedback effect through inhibin, which will act to reduce FSH secretion. The Sertoli cells also secrete that androgen binding protein that helps trap the testosterone in the lumen where these spermatozoa are maturing. And that brings us to the end of my presentation. I want to thank you all for listening. I found it to be a very educational and enjoyable process. Here is a list of all of the sources that I used, as well as locations for all the photos that appear in the presentation. Once again, thanks for listening, and I hope you enjoyed it.